Okay, well, hello everyone. Thank you for attending this talk today. My name is Brady West from the University of Michigan, Ann Arbor, and I'll be talking about measures of selection bias in regression coefficients estimated from non-probability samples. I'd like to acknowledge my team of co-authors, Rod Little, Rebecca Andridge, Phil Boonstra, Aaron Ware, Anita Pandit, and Fernanda Alvarado-Layton. This has definitely been a very collaborative project, and I'm excited to present this to you today. I'd also like to acknowledge support from a grant from the National Institutes for Health, an R21 grant. And I'd also like to acknowledge the National Survey of Family Growth, who enabled us to use these data, and clarify that the views expressed in this presentation do not represent those of NCHS or the other funding agencies. Finally, we'd also like to acknowledge the support of Gonzalo Abacasis from the Genes for Good study and David Weir, the PI of the Health and Retirement Study, who also enabled us to work with their data very graciously on this project. So our problem we want to talk about with this work is that many data collections are turning to the measurement of non-probability samples to save costs and potentially increase sample sizes. We've heard a lot about that at Bigsurv this year. This is true in survey research. It's also true in clinical trials, which often focus on volunteers, and genetics research as well, where GWAS often rely on volunteers. Unfortunately, the selection bias that's associated with these samples, which may be non-ignorable in nature, could affect estimates of interest based on the samples, whether those estimates of interest are descriptive, like means and proportions, or they're analytic, like regression coefficients in nature. So there has been prior work done on trying to quantify selection bias. Previous work presented at APOR and other conferences on survey research has developed measures of non-ignorable selection bias for means. There's a paper by Rod Little and colleagues, and also proportions, a paper led by Rebecca Andridge and colleagues, that are estimated from non-probability samples. So there has been work done on quantifying non-ignorable selection bias in non-probability samples for descriptive estimates. And simulation studies, a paper that's under second review by Phil Boonstra and colleagues, do provide solid evidence of the ability of these novel measures to actually detect selection bias. But many researchers are interested in studying relationships between variables, and no prior work has really developed similar measures of selection bias for estimates of regression coefficients that would be based on non-probability samples. So at that point, you might ask, well, what about the Heckman selection model? In this setting, without auxiliary variables or structural model assumptions, the data provide no information about regression coefficients for unselected cases that aren't part of the non-probability sample and how they differ from the coefficients for the selected cases. So we don't know anything in the data without this additional information. And for the Heckman selection model, which has been widely used in many different fields, selection is assumed to occur when there's a latent variable, let's call it L, that crosses a threshold. And L is assumed to have a joint normal distribution with the outcome of interest Y. So properly identifying the Heckman selection model requires unverifiable assumptions that ultimately exclude subsets of the predictors from the regression model for Y or from the model for L that ultimately determines selection. And Rod Little has done some work on this, talking about this limitation in past writings here. So our proposed approach is simpler analytically and computationally. And furthermore, unlike the Heckman selection model, it does not require having microdata for the non-selected cases in order to estimate the model. So we offer some advantages in that regard. So here's our setting. A non-probability sample has data available on Y, which is the outcome variable of interest, Z, which is a set of predictor variables that are of direct interest for a given research investigation, and then A, which are auxiliary variables that are not of direct interest for the current investigation, but are still related to Y after we condition on the predictors of interest Z. So these might be covariates in sociodemographic subgroup analyses or post-treatment outcomes related to that Y variable in clinical trials. We also have means, variances, and covariances of Z and A available for our target population, or we can estimate these means, variances, and covariances based on a probability sample that's describing our target population. Next, let X be the linear combination of A and the estimated regression coefficients for A based on a regression of Y on both Z and A fitted to the non-probability sample. So in other words, X is the best predictor of Y after we condition on Z. It's that linear combination of A and the regression coefficients that are estimated for A when we also have Z in the model. Again, we'll call that X moving forward. The model that we're proposing is a pattern mixture model for Y and X conditional on Z, given Z, where S is a binary indicator of selection for the non-probability sample. So here you can see this bivariate normal pattern mixture model for these variables X and Z. And notice that the probability of selection is written down as a function of X, Y, and Z, where this Y star combines X and Y and also introduces a phi parameter that defines the dependence of the probability of selection on Y. So this allows us to examine the extent of the non-ignorability and consider different scenarios where we toggle the value of that phi parameter to see how heavily selection depends on both X and Y. And we see a different pattern of means, variances, and covariances depending on whether you're selected or you're not selected based on the pattern mixture model. So we're defining indices of the selection bias in estimated regression coefficients based on that pattern mixture model. So under that model, one can show, and I'll point you to a paper in the references at the end, that the measures of unadjusted bias relative to the non-selected cases, so it's a little bit of a mouthful, but the MUBNS measures of unadjusted bias, notice that these quantify the differences between the intercept for the selected and the non-selected cases, and also the differences in the regression coefficients for the predictors of interest Z for the selected relative to the non-selected cases as a function of parameters defined by that pattern mixture model. What's important to note is that these measures are a function of phi, and that enables us to perform sensitivity analyses. So if we vary the values of phi, what do we get in terms of these measures that capture evidence of selection bias? The coefficients from the regression of X on Z, which are important for quantifying these indices for the non-selected cases, can be computed using the summary statistics for A and Z that are available for the target population. That's something else that we demonstrate in this paper. These indices are also a function of rho, which is the correlation of X and Y when we condition on Z, and the residual variances from the regressions of Y and X on X and Z. So the key point is we have all the information we need to compute these measures in this particular context. Some additional remarks on the proposed indices: we can actually drop the NS and compute measures of unadjusted bias relative to the population coefficients rather than the coefficients for the non-selected cases, simply by multiplying the MUBNS indices by the overall non-selection rate. And when we talk about non-probability samples, that usually approaches 1. If we're fitting models that only have an intercept, we end up with the same SMUB indices that were proposed in the paper by Little et al. For means. So there's consistency there as well. We can use fully Bayesian approaches to draw values of phi, that sensitivity parameter that defines the non-ignorability, and subsequently values of the indices to make inferences about the bias itself. So that's another nice feature. We can simply subtract the MUB indices or draws of the MUB indices, if we want to be Bayesian, from the coefficients that are estimated based on the selected sample to adjust our inferences for bias. So it's not just to quantify bias, we can use these approaches to adjust inferences as well. It is important to have these strong conditional correlations between X and Y, so having strong rho values is important. And extensions to non-normal variables based on this kind of model are also possible. And right now we're leaving that for future work. So our simulation study that we performed, some of the main findings, just to kind of summarize here, we looked at an in-depth simulation study to evaluate the performance of these MUBNS indices in a variety of scenarios. And we found that stronger conditional correlations between A and Y are important for improved performance as expected. So again, that defines that rho parameter that we were talking about. Setting phi to 0.5 and computing the indices tends to work very well in most scenarios. And when selection is a function of Y, MUBNS measures with higher phi values perform better, as we would expect based on the model. But setting phi to 0.5, which means that selection is a combination or a function of a combination of X and Y, tends to work well in different scenarios. Bayesian credible intervals for the MUBNS indices that are based on random uniform 0-1 draws of phi offer good coverage of the actual differences in coefficients across several different scenarios. And this good coverage of the credible intervals is not simply arising due to the intervals being extremely wide. So the intervals are reasonable as well, at the same time providing good coverage. So very promising results from some simulation studies that are again described in this paper by West et al. In 2020. So we looked at a couple different applications of this approach in a couple different contexts. The first one was to look at polygenic scores and their relationships with observed phenotypes in more of a genetic setting. So selection bias is an important concern in the volunteer samples that are often measured for genetic research. And polygenic scores, or PGSs, summarize the genetic risk of having certain values on phenotypes, things like weight, depression, education, based on the human genome. And the widespread use of these scores in current biosocial research assumes that these scores actually correlate with the phenotypes of interest. So this is an important assumption. And we wanted to evaluate this assumption using data on non-Hispanic volunteers who were ages 50 and above in the Genes for Good study. You can learn more about this study at genesforgood.org. These volunteers enroll via Facebook, then they take health surveys, and they mail saliva samples in for the genetic data collection. And we use the health and retirement study, which is a large probability sample of persons age 50 and above, as a benchmark, and to compute our summary measures that we needed for the indices. So for a variety of different phenotypes that were measured, we computed polygenic scores using the exact same genetic information in both Genes for Good and HRS. And then we regressed the observed values of the phenotypes Y on the computed polygenic scores for the Genes for Good sample, which was Z, and we estimated the bias in these coefficients using our indices. The auxiliary variables A that we used to form X included several sociodemographics and other phenotypes that were not considered in a given model of interest for that phenotype. And we used survey weighted estimates based on the HRS data to compute the summary measures for A and Z, which again were necessary inputs to be able to compute these measures. We then used the survey weighted HRS coefficients like population benchmarks for evaluation. So this table shows some important results from this first application, and we're highlighting these results in the table. The correlation of the MUBNS posterior medians, so when we used a Bayesian approach and we computed the posterior medians, the correlations of these medians with the actual estimated bias measures was about 0.56. So there's a pretty good positive correlation with the actual measures of bias based on the HRS coefficients. The measures also did a good job of detecting notable biases. So for example, highlighted in the table, the estimated intercept for height, there's a negative bias there. And the polygenic score slope for college degree, where the sign of that slope would completely flipped compared to GFG and comparing GFG and HRS. And our indices picked up on that flip. So this was promising. The intervals can also be very wide with low conditional correlations. We see an example of that for the BMI phenotype, but they can also work well for non-normal items. So like an indicator of having a college degree. So there are promising results here, but there are a couple of drawbacks, especially when that conditional correlation tends to be a little bit lower. Okay, so a second application, we looked at smartphone users in the National Survey of Family Growth, and we consider the scenario where you have a smartphone survey that tends to recruit individuals with less than high school education, who may have a higher response rate when incentives are offered. So we'll call this a hypothetical non-probability sample. We identified such individuals in the National Survey of Family Growth, and we treated the full NSFG like the known population of interest. Our Y variable was the number of months that they've worked in the past year. Our Z variables of interest were age, 3 different age groups, and then gender, whether you're male or female. And for our A variables used to form X, we use race, ethnicity, income, marital status, region of the US, current employment, and presence of children under 16 in the household. And again, these are variables where population information may be available, including means, variances, and covariances. Our descriptive objective was to regress the months that are worked in the past year on age and gender. So those were the two primary predictors of interest. And because we're using the NSFG as the kind of target of interest, we can actually compute the MUB index because we know the sampling fraction. So we can get rid of that NS extension. So here's some key results from the second application. The conditional correlation of X with Y was about 0.692 in this case. So that satisfies that, that necessary criterion for having good performance of the indices. The estimates from the smartphone sample suggest significantly lower mean past year employment for younger females and a larger male-female gap, okay, in that mean. And these biases were detected by our proposed indices. There was also a high correlation of estimated biases with the medians, again, of these MUB posterior distributions. So it seems like these medians do tend to work very well in the fully Bayesian approach in terms of indicating this bias, but more promising evidence of these indices being able to pick up on this kind of selection bias. So in summary, we've developed these simple model-based indices of selection bias for regression coefficients that are estimated from non-probability samples. We've evaluated the utility of these indices in different settings. The indices are effective when informative auxiliary variables are available, and Bayesian inference about the bias is also possible. We've provided easy-to-use R code with examples on our GitHub page, so you can see the link to our GitHub page here if you'd like to play around with the R code and computing these indices. There is a lot of methodological work that's still needed on polygenic scores and especially how they're computed. So we used a pretty plain vanilla version of the polygenic scores in this case. And again, we mentioned that future work needs to consider models for non-normal outcomes. And finally, you're welcome to email me at bwest@umich.edu with any questions about this presentation. And I would like to thank you very much for attending today. I'm hoping there will be some questions in the chat that we can talk about. Here are the references as well that we talked about in the presentation, and I'm happy to share any of these papers with you if you would like. So thank you very much again.